This is the SciComm 504 podcast. I'm Yuan Liu. The ocean covers approximately 70% surface area of the Earth. We fish in the ocean. We swim in the ocean. We sail in the ocean. Yet how much do you know about the ocean? Do you know that there are tiny photosynthesizers floating in the ocean? At some places, as dense as 1 million cells in a raindrop. These cells, what we call phytoplankton, are as important as terrestrial plants in sequestering carbon. As a matter of fact, they account for half of the carbon dioxide fixed as organic carbon each year. As vast and barren as it looks, the ocean is full of lives, especially really small ones. Although being small, these tiny cells are by no means trivial. Life in the ocean depends heavily on them. Each year, phytoplankton fixes tons of carbon into plant material, which is preyed upon by herbivorous microorganisms known as zooplankton. Examples are ciliates, dinoflagellates, and copepods. These small zooplankton are grazed upon by larger zooplankton. Krill, for instance. The krill are consumed by small fish like tuna, who eventually will end up in an even bigger fish's stomach, such as a shark. From the food chain described above, you see how important phytoplankton are. The ocean's capacity to take up carbon and regulate the global warming effect depends on these tiny cells. The picture of the microbial world in the ocean is still fuzzy. Being too small for direct observation and too difficult to grow in a lab are the main reasons for our lack of knowledge. Scientists have developed molecular techniques to study the microbes in the ocean without having to grow them in the lab. I am trying to identify which microorganisms are associated with one of the harmful algae, the brown-tide species in Great South Bay, New York. You know, the fingerprints are unique to each person. Similarly, I take fingerprints of the whole microbial community from the seawater using DNA information. The DNA fingerprints will reveal the secrets of who are the small cells co-occurring with the brown-tide species, and also how their relative abundance changes with the progression of the brown-tide bloom. Without having to identify species under the microscope, I want to understand what environmental factors are important for the pattern observed, and more generally, why we see what we see in that drop of water. Thanks for listening. This is Yuan Liu signing off from SciComm 504 at Stony Brook University.